Chapter 4, Parasitic Protozoa, Form, Function, and Classification. Form and Function, Part 3. When it comes to locomotion, okay, all these cells have to be able to move. They are not plants. They are not fungi that are sessile. These are want to be cells that are able to move because some of them, many of them are going to be predators. Okay, they are searching for anything smaller than them that they can ingest. If they are the parasitic forms of these protists, they are going to need to be able to swim through your gastrointestinal tract. They're going to need to be able to swim through tissues. So you're going to see that there will be some form of locomotion, ability to move. Three forms we see that are predominant. Pseudopodia. Okay, if you've ever seen video of amoeba moving, remember Pseudopodia means that way they, fake step, fake foot. They project out part of the cell and then the rest of the cell flows in. And we'll talk about that at the end of this lecture, more depth. We also have the undulating, what the textbook and many of the papers refer to as Angelopodia. These are the structures that undulate, that move, that allow for movement of the water, and you move enough water, you move the cell. First one is the flagella. Okay, the flagella is going to be important because this is the long protrusion of the cell. Keep in mind, flagella and cilia are still internal to the cell. They look like they're external appendages, but they are internal projections of the plasma membrane. When you look at the flagella and the cilia to a lesser extent, you're going to see that 9 plus 2, orientation of microtubules. They refer to this as the annexamine. This 9 plus 2 microtubules, that at the base of it is the kinetozone, okay? What you would refer to as the basal body in animal cells. Here in protists, they refer to as kinetozone. Why the different name for something that serves the same purpose? The structures are slightly modified. The kinetozome, more often than not, will be interacting with other organelles, specifically the mastodont system, which we'll talk about in a couple of slides. Cilia is also going to have its own version of an axoneme, keep wanting to put in an extra N there, axoneme, as well as basal bodies, kinetosomes. But here, since there's going to be more than one or two flagella, like you would see with protists, cilia, there's going to be hundreds, if not thousands of them, depending on the size of the cell, depending on how many, they're going to have there. They refer to this as the infrasiliature. So when you look here, again, you'll see that 9 plus 2 orientation. At the base of the flagella, you'll see the kinetozome. Okay. You'll see that with the flagella, for some of the genus species we'll be discussing, there's going to be this mastodont system. The mastodont system, its composition is going to be vary species to species. It is a complex of organelles that are associated with locomotion, energy production, and numerous other metabolic functions for the cell. Why can I not be more specific? Here's exactly what it does. Well, like I said, it depends on the species. We also have some issues with trying to study these, because to study them means we have to modify it. If we change this, how does that affect other metabolic functions, blah, blah, blah. Have to be able to isolate the different components. Isolate them so that we can study them, see, what enzymes, what proteins, things like that are present. Well, when we go and try to isolate these, we disturb the whole process there. So we're not always sure what the microenvironments are between these. We see that for these mastodont systems, they could have those dictosomes that we talked about in a previous lecture. Those are those modified Golgi apparata that we see with some protists. There will be other compositions, all we know, generalized when it comes down to it, mastodont system's going to have a flagellum, 1, kinetozome or 2, and then numerous other organelles that could be there. Maybe there's a dictosome. Maybe there's mitochondria. Some are going to have what is referred to as the costa. The costa is a long, striated rod made of different proteins. What's its function? Don't really know. Some ideas that it's there to act as a stabilizing foundation, because you got the flagella moving, and you got to have something more than just a kinetosome, give it something to move against, or it's going to be flopping all over, which is going to be completely disrupting the cell. Could also be a place for anchoring of enzymes and whatnot. So if you don't have a mitochondria, then you can have the costa and some of these other organelles that associate with the mastodont system can be a place to anchor, to house the different enzymes that we would associate with the mitochondria and oxidative phosphorylation. And when we look at the cilia, we see that it's not just, the kinetosome, the basal body at the base of all the cilia, and they're 9+2 exoneme. What you're going to have is what they call kinetids. Kinetids are going to be parabasal bodies. They're going to be microtubule structures, as you see here in this figure right here, that form this meshwork that underlie all the cilia, so that it gives it a strong foundation, so that when you get these cilia moving, you're not getting the whole cell undulating because the cilia are moving. No, that meshwork of microtubules gives a strong foundation so that the cell body itself, the membranes and all that, aren't fluctuating because the cilia are in movement. You're also going to see, if you look closer at this figure, was a 4.6 in the textbook, you'll see that there is other organelles that will be associated. So you have the pellicle, going to have the membranes below it, the alveolar sacs, and then you're going to have the plasma membrane. And depending upon which one of the species you're talking about, intersposed in the alveolar sacs could be some modified mitochondria or just below the plasma membrane, producing ATP that's going to be used up because these cilia are constantly in motion. Okay, and it's not just a couple of them. Remember, it's going to be hundreds, if not 1,000 or so, cilia all around 360, all around the cell, in constant motion. These kinetids, as they're referred to, can be singular, dual, dikinetids, trikinetids. That's how many of these different structures you're going to find associated at the base of each of the cilia, providing stability, providing energy, providing whatever is necessary to keep that cilia, that individual cilium, maintained. What's also interesting is that there's the cilia that wrap around the cell, and then there's going to be a modified versions of them around an oral opening. Okay. When I say oral opening, I'm not saying that these guys have a mouth. What they have is an area, as you see here in this artist rendition, that's referred to as an oral groove. It's an indentation in the pellicle. The pellicle has been modified. It's thinner, maybe even non-existent at this area. The membranes, the plasma membranes, and all the accessory membranes are also modified. The cytoskeleton just below it, the microfilaments and all that, are modified so that this area is promoted, is altered, changed, and is more than ready to phagocytize. Because remember, the rest of the cell is covered by that somewhat firm, somewhat solid pellicle, which is not going to allow for things to pass through, large things. So by having this oral ciliature, the cilia, instead of moving in the pattern that all the other cilia are, which are to move the cell through the water, these cilia, their beating pattern is to waft water and whatever things are floating in that water, bacteria, fragments of smaller protist, whatever, into that oral groove so that uptake, phagocytosis, can occur. And then that pseudopodia that I mentioned at the first slide of this lecture. Pseudo, fake, podia, foot, the fake step. Well, it turns out that it's more than just what we've seen with amoeba, where that one part of the cell extrudes out and then the rest flows in. Turns out that's more, to the correct terminology for that is lobopodia. A lobe of the cell extends out, the rest of the cell flows in. There's also philopodia, rhizopodia, axopodia. And you look at these figures here, these artist renditions, shows you the different protrusions, the way that the cell can flow out. Why do you have to have these modified versions of it? Why can't you just have the lobopodia like you see with the amoeba? Well, the pellicle. Amoeba don't have a pellicle. Amoeba don't have that thick, semi-rigid shell. Many others do. So they have to have some way to protrude out the plasma membrane and allow it to move. So the rest of the cell either flows in or it protrudes out and it moves, waves around, and allows the cell to somewhat swim. How's that different than the flagella? Well, the flagella has the 9+2 axoneme. When you look at the other, especially the two at the bottom here, they don't have the 9+2 in their protrusions. Those protrusions are just microfilament based. So the plasma membrane gets pushed out by the actin filaments, and then it just kind of moves and is there, and it's just kind of moving like this, not the nice wave-like pattern you would see with flagella and cilia.